welcome to the deep dive. Ever wondered how psychologists actually figure out why we do the things we do? Or, how our minds work? Today, we're sort of pulling back the curtain on psychological research methods.
Yeah.
Think of this as your shortcut, maybe, to understanding the science behind the headlines and, well, the studies you hear about.
Exactly. Our goal really is to give you a solid sense of how psychologists investigate behavior, mental processes.
Right.
So you can be, well, a more informed consumer of all that psychological info out there, knowing what's based on solid evidence and what might be, a bit more speculative.
Okay. We've got a great set of resources lined up. We'll be looking at some pretty clear explanations of stats and surveys from YouTube, along with a really deep dive into an open access textbook. It covers the whole scientific approach, ethics, different ways to design studies.
How we measure thoughts and feelings, the basics of statistics,
And even some of the tricky bits, the pitfalls researchers need to watch out for. Okay, so let's get right into it. What makes psychology a science? It isn't just about opinions, right?
Oh, not at all. No. The foundation is something called systematic empiricism.
Systematic empiricism. Now,
Yeah. So imagine you have a hunch about something, say, like, people are happier when it's sunny. Instead of just noticing a few happy people on a sunny day and saying, see, proof.
Yeah.
Systematic empiricism means you'd actually plan it out. You'd observe lots of people, record their mood in different weather, and then analyze that data carefully.
Ah, okay. Planned observation.
Exactly. The textbook actually mentions a study where researchers counted how many words people spoke to test assumptions about who talks more.
Oh, right.
And it turns out our casual observations, they can be way off.
So it's about moving beyond what we just think is true, our sort of folk psychology, and actually getting solid evidence. The sources point out some classic examples where common sense kind of fails, right? Like the idea that punching a pillow helps with anger.
Right, or the myth that we only use, 10% of our brains?
Yeah, that one.
Precisely. And while, our intuitions aren't always wrong, the textbook really emphasizes that having these systematic methods is crucial. It's how we get a reliable handle on behavior and mental processes, avoiding our own biases.
Okay, makes sense. So psychologists have this scientific mindset. How do they then go from that broad approach to asking specific research questions? What sparks their curiosity scientifically?
Well, it starts with framing questions that you can actually test through observation and data, empirically testable questions.
Right, so you have to...
And what makes a question interesting to the scientific community often comes down to a few things. Is the answer, currently unknown or debated?
Okay.
Does it fill a gap in what we already understand? Or maybe does finding the answer have practical, real-world benefits?
That makes a lot of sense. It's not just any random thought. It has to potentially lead somewhere new. And the sources talk about phenomena versus theories. What's that distinction?
Okay, so think of a phenomenon as a result, something that's been found again and again in research. It's pretty established.
Like what?
The finding that expressive writing about your feelings can actually have positive health effects, that's a phenomenon.
Got it.
A theory, though, is the explanation for that phenomenon. It's the proposed reason why expressive writing might help. It's the why behind the what.
Right. Phenomena are the reliable whats, theories are the whys. So how do researchers test those theories?
Often, they use what's called the hypothetico-deductive method.
Okay.
Sounds fancy, but the idea is you start with a theory. From that theory, you derive a specific testable prediction. That's your hypothesis.
A specific prediction.
Yeah. Then you design a study to test that hypothesis. You collect your data, see if it supports the prediction, and based on that, you evaluate your original theory. Maybe you refine it. Maybe you find evidence against it.
Like a cycle.
Exactly. The textbook uses that great example of social facilitation.
Oh, right. The idea that having others around affects performance.
And how researchers like Zhang tested this with, even cockroaches in mazes.
The cockroach study. I remember that. Didn't they find that cockroaches ran faster through simple maze when others were watching?
Yes.
But they actually did worse on a complex maze with an audience.
Exactly. Zhang's work is a perfect illustration. The theory led to specific predictions, and the results either supported or challenged parts of the explanation. It also shows how different perspectives or models can sometimes explain the same thing.
Okay. Now, before any of this testing happens, before a study even gets off the ground, there's something absolutely critical: ethics.
Oh, absolutely essential, non-negotiable. The sources mention several really important ethics canons that shaped modern research, things like the Nuremberg Code, the Declaration of Helsinki, the Belmont Report.
These came out of some, pretty problematic historical studies, didn't they?
Sadly, yes. They evolved because of past research that raised serious ethical alarms. In psychology specifically, the American Psychological Association, the APA, has its own detailed ethics code.
Wow.
And it covers everything from getting informed consent, how to handle deception responsibly if it's absolutely necessary, the ethical treatment of animals, and just maintaining scholarly integrity. It's all about protecting the rights and well-being of participants.
And to make sure these codes are actually followed, institutions have IRBs, right? Institutional Review Boards.
Exactly. If an institution gets federal funding for research, it needs an IRB. These are committees that review every research proposal before it starts.
What do they look for?
They weigh the potential risks to participants against the potential benefits of the knowledge gained. A huge part of their job is ensuring proper informed consent.
Okay, informed consent. What does that really mean? It's more than just signing a form, isn't it?
Oh, much more. It means researchers have to give potential participants enough information about the study, what they'll do, any possible risks, any benefits, confidentiality, so they can make a truly informed choice about participating. It has to be voluntary, and they need to understand what they're agreeing to.
So transparency is key. What about situations where researchers feel they need to withhold some information or even use deception? That sounds really tricky ethically.
It is tricky, and it's handled very carefully. The APA Ethics Code only allows deception under really strict conditions.
Like when?
Like when the potential scientific value justifies it, and there's no other way to study the phenomenon, and participants aren't expected to be harmed. Crucially, if deception is used, participants must be fully debriefed afterwards.
Debrief.
Yeah, that means explaining the true nature of the study, why the deception was necessary, and addressing any misconceptions or negative feelings it might have caused. The default is always to minimize risks and avoid deception if possible.
And it's not just about human participants, right? Animals in research.
Absolutely. Research with animals is also governed by strict ethical guidelines and regulations. The focus is on humane care, proper housing, minimizing any pain or distress, and ensuring the research is scientifically justified.
Okay, so we've got our ethical house in order. Now, how do we actually measure the things psychologists are interested in, like, happiness or memory or intelligence? You can't just put a ruler to those things.
Right. That's where the idea of psychological constructs comes in.
Constructs.
Yeah. Things like intelligence, anxiety, self-esteem. These are characteristics or tendencies that aren't directly observable. They're concepts. Psychological measurement is the whole process of assigning scores or numbers to people to represent these constructs.
How do we do that?
We use different types of measures. We could ask people directly, like using a questionnaire that's self-report, or we could observe their behavior in a certain situation, behavioral measures. Or we could even measure physiological things like heart rate or brain activity.
So trying to capture these intangible things with numbers or observations, the textbook talks about operationally defining a construct. What's that about?
Yes. Operational definition. It just means defining a construct very specifically in terms of how you're going to measure it in your particular study.
So less abstract, more concrete.
Exactly. If you're studying anxiety, your operational definition might be the score on the Beck anxiety inventory, or the number of times a person fidgets during a public speaking task. It makes the abstract concept measurable for that specific research.
Okay. So we have a way to measure it. But how do we know if our measurement is actually any good? That gets into reliability and validity, doesn't it?
Precisely. Two crucial concepts. Reliability is about consistency.
Consistency.
Yeah. Imagine stepping on your bathroom scale 3 times in a row. If it gives you wildly different weights each time, it's not reliable.
Right.
In psychology, retest reliability means if you give the same measure to the same person at 2 different times, assuming the thing you're measuring hasn't actually changed, you should get similar scores.
Okay. And there's another type, internal consistency.
Right. Internal consistency is about whether the different parts or items within a single measure are all pulling in the same direction, measuring the same underlying thing. Like on a self-esteem scale, all the questions should generally relate to how someone feels about themselves.
Makes sense. So that's reliability, consistency. What about validity?
Validity is arguably even more important. It asks, is this measure actually measuring what it's supposed to be measuring?
Ah, the accuracy question.
Exactly. And there are different facets to validity. Face validity is just, does it look like it's measuring the right thing on the surface?
Seems basic, but probably useful.
It is. Then there's content validity. Does the measure cover all the important aspects of the construct? Criterion validity is about how well our measure correlates with other established measures of the same thing, or with relevant outcomes.
So, if my new depression scale correlates well with the doctor's diagnosis,
That would be good evidence for criterion validity, yes. And then there's discriminant validity, showing that your measure doesn't correlate strongly with measures of things it shouldn't be related to.
like an intelligence test shouldn't strongly correlate with, say, shoe size.
Exactly. It helps confirm you're zeroing in on the right construct.
The textbook also brings up Stevens's levels of measurement: nominal, ordinal, interval, ratio. Why do researchers need to worry about these?
The levels. They basically describe the nature of the numbers you're using. Nominal is just categories, like political party affiliation. Ordinal involves ranking, like first, second, third place.
Okay.
Interval scales have equal intervals between the numbers, like temperature and Celsius, but no true 0.0 degrees doesn't mean no temperature. Ratio scales have equal intervals, and a true zero, like height or weight zero, means none.
And why does this matter?
It matters because the level of measurement dictates what kinds of math and statistics you can properly use on your data and how you can interpret the results. You can't calculate a meaningful average of nominal categories, for instance.
Got it. Okay, let's shift gears to how studies are actually designed. There's this big split: experiments versus non-experiments. What's the fundamental difference?
The absolute key to a true experiment is manipulation and control. The researcher actively manipulates at least one variable, the independent variable, to see if it causes a change in another variable, the dependent variable.
Manipulation.
Fun.
So they're actively changing something for different groups.
Yes. And they try to control other factors, extraneous variables that might mess up the results. Often they'll also use random assignment to put participants into the different conditions or groups. It's this combination of manipulation, control, and random assignment that lets experiments make claims about cause and effect.
Okay. And in a between subjects experiment, each person is just in one condition.
That's right. One group gets treatment A, another group gets treatment B, maybe a third gets a placebo, or nothing, the control group. The trick is making sure those groups are really similar to begin with, which is where random assignment helps.
Right. So you can assume any difference at the end is likely due to the treatment.
Ideally, yes.
Yeah.
The textbook also mentions field experiments, which happen out in the real world, not a lab, can be more realistic but harder to control everything.
What about the other main type within subjects experiments?
In a within subjects design, the same people experience all the conditions. So the same group might try studying with music, then without music, and you compare their performance under both conditions.
So each person serves as their own control.
In a way, yes. It can be very efficient, fewer participants needed sometimes, but you have to worry about things like order effects. Maybe doing the first task affects performance on the second just because of practice or fatigue.
Right. The sources also stress standardized procedures and avoiding experimenter expectancy effects. What are those?
Standardized procedures just mean you treat everyone within a given condition exactly the same way, following a strict protocol. Minimizes variability.
Okay.
Experimenter expectancy effects are more subtle. It's when the researcher's own hopes or expectations about the study outcome unintentionally influence how they treat participants or even how they observe behavior.
So if I expect the new drug to work, I might unconsciously be friendlier to patients getting it.
Exactly. Or you might interpret ambiguous results in a biased way. To combat this, researchers use blinding. Single blind means the participant doesn't know which condition they're in.
Oh.
Double blind means neither the participant nor the researcher interacting with them knows who is in which condition. That's considered the gold standard for minimizing expectancy effects in many trials.
Okay, that makes sense. Now, flip side, non-experimental research. What defines that?
Simply put, non-experimental research lacks either the manipulation of an independent variable or random assignment to conditions, or both.
And the big consequence is?
You can't draw firm cause and effect conclusions. You can observe relationships, you can describe situations, but you can't say for sure that A caused B.
And correlational research fits here, right? Looking for statistical relationships between variables. But the big mantra, correlation does not equal causation.
Absolutely fundamental. Just because two things go together like ice cream sales and crime rates doesn't mean one causes the other.
Right, they both go up in the summer because of the heat, presumably. The third variable problem.
Exactly. That's a third variable problem. Or maybe the direction of causality is reverse the directionality problem. The textbook uses the exercise and happiness example. Maybe exercise makes you happy, or maybe happy people are more motivated to exercise, or maybe good health leads to both. Correlational data just shows the link, not the why or the direction.
How do researchers gather correlational data?
Lots of ways. Observing behavior in natural settings, naturalistic observation, using existing records or data sets, archival data, and of course, surveys.
And we also have qualitative research, which sounds like a totally different approach.
It often is, yeah. Qualitative research dives deep into understanding people's experiences, perspectives, and the meanings they make. It usually involves non-numerical data.
Like interviews.
Exactly. Open-ended interviews, focus groups, detailed case studies. The textbook mentioned a study using unstructured interviews to really explore women's experiences with postpartum depression in depth. The goal isn't usually statistics or causation, but rich description and interpretation.
Sometimes research questions are more complex, needing more advanced designs. The sources touch on these briefly. What's the point of, say, using multiple dependent variables?
Well, it just gives you a richer picture. Instead of just seeing if a therapy reduces anxiety symptoms, 1DV, you might also measure quality of life, social functioning, maybe even physiological stress markers. Multiple DVs give a broader view of the effects.
And what about manipulation checks?
Those are important in experiments where you're trying to manipulate someone's internal state, like their mood or level of stress. A manipulation check is basically a measure you include just to make sure your manipulation actually worked.
So if I try to make people feel happy by showing them comedies, I might ask them afterwards how happy they feel just to check.
Exactly. Did the comedy actually make them happier than the control group? It confirms your independent variable was implemented successfully.
Okay, let's focus specifically on survey research for a bit. The YouTube stuff highlighted its reliance on self-report from a sample.
That's the core of it, yes. You ask a sample of people questions to get a snapshot of the attitudes, beliefs, or behaviors of a larger population.
And the key is getting a good sample.
Absolutely crucial. Ideally, you want a large, random sample. Random means everyone in the population has an equal chance of being selected. That's how you can generalize your findings back to the whole population with some confidence. And surveys can be done in lots of ways, in person, phone, mail, and obviously a lot online now.
Answering survey questions seems simple, but the textbook describes it as a cognitive process. What's involved?
Yeah, it's not passive. First, you have to interpret the question, what is it really asking? Then, retrieve relevant information from your memory. Then, form a judgment based on that info. Then, format your judgment onto the response scale, provided like a 1 to 5 rating. And finally, you might even edit your response, maybe to sound more socially desirable, before you actually give it.
Wow, okay. A lot going on. That must mean writing good questions is really important.
Hugely. The textbook mentions the Bior-Russo model as a guideline. Questions should be brief, relevant, unambiguous, specific, and objective.
Bior-Russo.
Okay.
And definitely avoid those double-barreled questions, right? The ones asking two things at once.
Oh, yeah. Those are terrible. Like, do you think the campus needs more parking and better food options? How do you answer that if you agree with one part but not the other?
Right. And the type of scale matters, too. Literature scales, numerical ratings.
Yes. And whether the scale is balanced, where you actually strongly disagree to strongly agree, or unbalanced. Even how the questionnaire looks, the order of questions, it can all influence responses.
And getting back to sampling, the textbook talks about different probability sampling methods like simple, random, stratified, cluster. These are ways to try and get that representative sample.
They are, but even with good methods, you can run into issues like sampling bias, especially non-response bias, where the people who don't respond are systematically different from those who do. Researchers need to think about how to minimize that.
Okay. Now, sometimes researchers want to focus really intensely on just one person, or maybe a couple of people. That's single subject research.
Exactly. Instead of averaging across large groups, you study a small number of participants very intensively over time.
How does that typically work?
Common designs involve establishing a baseline, measuring the behavior of interest before any intervention. That's the A phase. Then you introduce the intervention or treatment, the B phase, and keep measuring.
So an A-B design.
Right. But to be more confident the intervention caused the change, researchers often use reversal designs, like ABA. You go back to baseline, remove the intervention, and then reintroduce it. If the behavior changes systematically with the introduction and removal of the intervention.
Then you have stronger evidence it's the intervention doing the work.
Precisely.
How do you analyze data from just one or two people? You can't run the usual group statistics, can you?
Not typically, no. The main method is visual inspection. You graph the data across the phases and look for clear changes in the level of the behavior, The trend, is it increasing or decreasing, and the consistency within each phase. Are the changes convincing just by looking at the graph?
So it's more about visual evidence.
Primarily, yes. Though researchers might supplement that with simple stats, like calculating the mean score in the A phase versus the B phase, or something called the percentage of non-overlapping data, PND, which tries to quantify the difference between phases.
What are the pros and cons of this approach versus group research?
Well, a big strength is showing that an intervention works for a specific individual, which is really important in clinical settings. It's also good for studying rare conditions where you can't find large groups. The main weakness is external validity. It's harder to know if the findings will generalize to other people.
So they're kind of complementary. Group research tells you about averages. Single subject tells you about individual responses.
That's a great way to put it. They answer different but equally valuable questions.
Okay, so we've done our research experiment, survey, single subject, whatever. Now we need to tell people about it. That's where APA style comes in.
Yes, APA style. It's the standard format used in psychology and many other social sciences for writing research papers and citing sources.
Why have a standard style?
Clarity and consistency. It makes it easier for readers to know where to find specific information, the methods, the results, the discussion. It standardizes how we report statistics, create tables and figures, and list references so everyone's on the same page.
The textbook lays out the basic parts of an APA-style article: title page, abstract, the summary, intro, method, results, discussion, references.
Exactly. And it gives detailed guidance on how to format those references for journal articles, books, etc., how to present stats clearly in the text, and how to make effective tables and graphs like histograms or scatter plots. It's the common language of scientific communication in the field.
Okay, let's talk more about those statistics. We need ways to summarize all the data we collect. Descriptive statistics. The YouTube stuff gave a good overview of central tendency.
Right. Descriptive stats are just tools to summarize and describe the main features of our data. For a single variable, you might start with a frequency distribution. How many times did each score occur? Or draw a histogram to see the shape of the distribution. Is it bell-shaped? Skewed? Does it have one peak, unimodal, or two, bimodal?
And then we want a sense of the center of the data, measures of central tendency. The mean is the average, right? Add them up, divide by how many?
Yep, the mean, very common. But as the YouTube video rightly pointed out, it's really sensitive to extreme scores or outliers. One huge score can pull the mean way up.
So what are the alternatives?
We also look at the median, the exact middle score when you line them all up. It's not affected by outliers. And the mode, the most frequent score. Depending on the data shape, the median or mode might give a better sense of the typical score than the mean.
Okay. Besides the center, we also need to know how spread out the scores are. Variability.
Crucial. Are the scores all clustered together, or are they really spread apart? The simplest measure is the range, highest minus lowest, but it only uses two scores. Better measures are the variance and its square root, the standard deviation.
Standard deviation, that tells us.
It tells us, on average, how far each score tends to be from the mean. A small standard deviation means scores are tightly packed. A large one means they're more spread out.
And if we want to describe the relationship between two variables?
A scatter plot is the go-to visual. You plot each person's score on both variables. You can see if there's a pattern linear, curved, no relationship.
The textbook also introduces specific numbers for effect size. Cohen's d for differences between means.
Right. Cohen's d gives you a standardized measure of how big the difference is between two group means in standard deviation units. It helps interpret whether the difference is small, medium, or large, regardless of the original scale.
And for correlations, there's Pearson's R.
Pearson's R measures the strength and direction of a linear relationship between two variables. It ranges from -1, perfect negative correlation, to +1, perfect positive correlation, with 0 meaning no linear relationship. Like Cohen's d, there are conventions for interpreting R as small, medium, or large.
But the textbook cautions these only capture linear relationships, and things like restricted range can distort R.
Important caveats, yes. Effect size measures like D and R are super important because they tell us about the practical significance or magnitude of a finding, not just whether it's statistically unlikely to be due to chance.
Which brings us neatly to inferential statistics, using our sample data to draw conclusions or inferences about the wider population. The textbook explains null hypothesis testing.
Right, inferential stats help us generalize. Null hypothesis testing is a very common framework. You start by setting up two competing hypotheses. The null hypothesis, usually stating there's no effect, no relationship, no difference in the population, and the alternative hypothesis, stating there is an effect.
So you assume nothing is going on until proven otherwise.
Kind of, yeah. You collect your sample data and then calculate the probability of getting data like yours, or even more extreme, if the null hypothesis were actually true. That probability is the p-value.
The famous p-value, and the textbook really stresses, the p-value is not the probability that the null hypothesis is true, nor the probability that the alternative is true.
Absolutely critical point. It's the probability of the data, or more extreme data, assuming the null is true. A small p-value suggests your data would be surprising if the null were true, potentially leading you to reject the null in favor of the alternative.
What influences the p-value?
Two main things. The size of the effect in your sample, bigger effects lead to smaller p-values, and the size of your sample, larger samples also lead to smaller p-values, all else being equal.
But statistical significance, usually defined as p.05, doesn't automatically mean the finding is important or meaningful, right?
Exactly. You can get a statistically significant result for a tiny, trivial effect if your sample size is massive. That's why looking at effect sizes, like Cohen's d or Pearson's r, is so important alongside the p-value. Significance versus practical importance.
And there's always a chance of making errors with these decisions.
Always. A type I error is rejecting the null hypothesis when it's actually true, basically, a false positive, finding an effect that isn't really there in the population.
Okay.
A type 2 error is failing to reject the null hypothesis when it's actually a false negative, missing an effect that is there. Researchers also think about statistical power, which is the probability of correctly rejecting a false null hypothesis, avoiding a type 2 error.
The textbook also mentions some criticisms of this whole null hypothesis testing framework, like the arbitrary 0.05 cutoff.
Yeah, there's ongoing debate about its limitations. The file drawer problem, where non-significant results often don't get published, and whether focusing solely on rejecting the null is the best approach. It also introduces the one-sample t-test as a basic example of comparing a sample mean to a known population value using these principles.
Lots to consider when interpreting results. Statistical tools are useful, but definitely not infallible.
That's the perfect summary. Understanding the tools and their limitations is key.
Well, we have definitely taken a deep dive today into the core principles of psychological research methods.
Wow.
We've covered a lot from the scientific approach in ethics.
Measurement, different designs, experimental, non-experimental, surveys, single subject.
APA style for communication, and then descriptive and inferential statistics with all their nuances.
Yeah, and hopefully you, our listener, now have a much better feel for the systematic ways psychologists try to tackle the huge complexity of human behavior and the mind. It really relies on careful observation, thinking critically, and sticking to those ethical guidelines.
And definitely always remembering that correlation isn't causation, and statistical significance isn't the whole story.
Absolutely. So as you come across psych studies or news reports about behavior, maybe think about some of the things we discussed. Are the claims based on solid systematic evidence? Are the conclusions appropriate for the type of study done?
And maybe what further questions pop into your mind now about how we actually come to understand ourselves and each other? Something to chew on. Thanks for joining us on The Deep Dive.